In the other video, we learned about the cell cycle and we focused on interphase. The nucleus and the nuclear envelope are distinct, and the chromosomes are in a loose, thread-like conformation called chromatin. We talked about G1, S, and G2 taking place, and we said that 90% of the cell cycle time is spent in interphase. Now, our cells are what we call diploid. That means that we have 2 copies of every chromosome. In this case, in this image, there are 3 chromosomes, 1, 2, 3. And since we have 2 copies of each, 1 from a biological mom and 1 from a biological dad, we say that they are 2N. That means that each of the homologous chromosomes have the same genes, A and A represent the same gene in the same location. The alleles might be different, like here, or they might be the same, like here. But that's why we call them homologous. Same genes, same locations. So we're going to take this chromosome and we're going to look at what happens. So right now you have 1 chromosome. And then that is going to be duplicated in the S phase. So in S phase, you replicate that and you get the same exact identical genetic information. This is still considered 1 chromosome. It's just now replicated. So it's held together by a centromere, and each one of these arms is called a sister chromatid. So something else that happens during S phase is the centrioles. Okay, those are these organelles. They are going to be duplicated, and they always come in these pairs. So the pairs are duplicated. So once the cell leaves G2, mitosis can begin. So mitosis is the 10% of the cell cycle time where the cell is actually going through division. And the first stage of mitosis is prophase. Here, the centrioles that had replicated in the S phase start to migrate to the poles. They start to send out microtubules that we call spindle fibers. And the nucleus starts to dissolve. And the chromosomes, they start to condense so that you can kind of visualize them. Now, what ends up happening is the reason that this nucleus starts to dissolve is because the chromosomes can't move if the nuclear membrane is intact. The DNA is just too big. So this nucleus has to completely go away. In the stage between prophase and metaphase, we sometimes call that prometaphase. And what happens here is the spindles start to attach to the kinetochore. And the kinetochore is an attachment site for these spindle microtubules. So these spindle fibers attach to this kinetochore piece. The centromere, that's this region where the kinetochores are found. In this image, you can see that the centrioles are starting to send out their spindle fibers and they're starting to connect to the kinetochores. Then you've got metaphase, and all the chromosomes are going to move because the spindle fibers are attached, and the chromosomes get lined up at the center of the cell at the equatorial or the metaphase plate. They all line up. Then you've got anaphase, and that's where the chromosomes are pulled apart and they start to move to the poles. Remember, you've got that duplicated chromosome. So on our other image, that was G here and G there. So they're duplicated and they're the same on each side because they were replicated during S phase. And all those genes that were replicated, they're replicating faithfully and equally on each side. So those are what the spindle fibers start to pull apart. The spindle fibers start to pull these replicated pieces apart. Okay? And basically they're pulling the sister chromatids apart. They go to the opposite sides. And in this way, each pole is getting the identical copy of every single gene. In telophase, all that happened in prophase is reversed. The chromosomes start to relax, the nucleus starts to reform, and the spindle fibers start to break apart. Then you get to cytokinesis. In animal cells, you've got a band of microfilaments made of myosin 2 and actin, and they start to form a contractile ring at the former equator. Then it's going to go ahead and contract more and more until the cell begins to cleave in two, and we call that the cleavage furrow. And in a plant cell, you're going to go ahead and build up a cell plate made of cellulose. So you're just going to go ahead and build that cellulose up, and you build a cell wall where that equatorial plate was. And in any case, whether it's a plant or an animal cell, you now have two diploid daughter cells. They're going to be identical to what you had started with. Here's a quick overview. Feel free to stop the video and just go ahead and look through it. I like this bottom picture simply because it shows this idea that you are starting with two separate chromosomes, and those go ahead and replicate, and then those chromosomes are pulled apart. So you started with a 2N cell, and you ended with two cells that are both 2N or diploid. Again, keep in mind, this is one chromosome, and this is one chromosome. The only difference is this one is replicated. And then here you go. If you want to look at this, see if you can work through each of the phases and review some of the characteristics of each one for this plant cell, and see if you can work your way through mitosis.